Okay, welcome everyone to the second afternoon session. We're going to start this session with a talk by Chrysothemis Brown from the Memorial Sloan Kettering Center in New York. Chris is interested in antigen-presenting cells, and she has contributed to our understanding of the diversity of these cells in the immune system. Thank you very much for coming, Chrissy.
And thank you, Carlos, for the invitation. It's been a fantastic meeting. So as Carlos mentioned, in the lab, we're very interested in understanding how the immune system balances inflammatory responses to pathogens and tolerogenic responses to harmless antigens, such as tumor-associated antigens or commensal or dietary antigens. And we really focus on the antigen-presenting cells, which provide the cues that instruct different T-cell fates. And we heard very nicely from Pierre and others today about this division that occurs whereby different dendritic cells can provide different signals to drive the T-cell differentiation. But the one thing that has always remained a little bit enigmatic is the antigen-presenting cell that drives regulatory T-cell differentiation. And we know that the balance between tolerance and immunity is particularly critical in certain tissues. So obviously in the tumor, regulatory T cells are known to suppress immune responses to tumor-associated antigens. And that's obviously detrimental to the host. But in the intestine, you have this fundamental requirement for these peripherally induced regulatory T cells to suppress harmful responses against the microbiota as well as dietary antigens. And these peripherally induced regulatory T cells can be distinguished by expression of this transcription factor, raw gamma T. So we really wanted to understand how dendritic cells or antigen-presenting cells might drive the differentiation of peripheral Treg cells. And what we did know is that the requirement for immune tolerance is not static over the lifetime of an organism. And so that gives us some clues about how to unravel the mechanisms that underlie peripheral Treg induction. So a number of previous studies have demonstrated that there's this window of opportunity for generating tolerance to the gut microbiota. And so babies are colonized with trillions of bacteria as soon as they pass through the birth canal. And then you can see that they have bacteria all over their barrier surfaces from early life. And if you fail to establish tolerance to the microbiota, you're at risk of inflammatory diseases later in life, not just intestinal diseases, but also allergic diseases. And so what previous studies had demonstrated that there's this window of opportunity for establishing tolerance, and that underlying that window is the development of these immunosuppressive regulatory T cells. And so if you transfer naive T cells into a young mouse around 2 weeks of age, they're much more likely to differentiate into regulatory T cells than if you transfer naive T cells into an adult mouse. And so really what we find is that these regulatory T cells start to increase around the time of weaning, and then they plateau within the intestine. And so that suggested to us that there might be something unique about the antigen-presenting cell niche within the intestine that would drive the differentiation of these cells. And so I'm only briefly going to summarize this because it's been published, but through our study of early life antigen-presenting cells, we identified this new lineage of antigen-presenting cells that are distinguished from dendritic cells by expression of this transcription factor, raw gamma T, which provides a nice symmetry with the raw gamma T expressing Treg cells. And what we showed is that there's 4 distinct subsets, but one of the subsets expresses this integrin beta 8, which activates latent TGF-beta, which drives the differentiation of naive T cells into regulatory T cells. And I'm just going to highlight a few interesting aspects of the cells that will be relevant to the rest of the talk. So I think perhaps the most exciting thing for someone that sort of works on early life immunity is that they have this very interesting developmental wave. So they peak in gut lymph nodes just at the time that you're weaning mice from milk to solid food. So this is obviously the time that the microbiota undergoes this huge diversification. And it's also the same time that you're introducing dietary antigens. And those two phenomena are actually linked to each other. And you can see that this wave of Thetis cells actually precedes the development of regulatory T cells within the intestine. And the second interesting thing, which is how the cells actually got their name, is that when we looked at the transcriptome of the cells, they had a very interesting feature, which is they not only have a transcriptome that overlaps with dendritic cells, in particular these migratory dendritic cells, but they also have overlapping transcriptomes with medullary thymic epithelial cells. And MTECs are a very interesting population of cells. They're obviously not thought of as canonical antigen-presenting cells in that they're non-hematopoietic, but they play important roles in the thymus in directing tolerance through the generation of thymic regulatory T cells as well as deletion of autoreactive T cells. And that process is mediated by the expression of this nuclear factor, AIR, which drives promiscuous self-antigen expression. And what was very interesting is that amongst the cells we identified, 2 of the subsets expressed AIR. And so we named them Thetis cells because of this hybrid phenotype where they overlap both dendritic cells and medullary thymic epithelial cells. Now, interestingly, although they have transcriptional similarities to dendritic cells, they actually arise from a distinct progenitor. So dendritic cells arise from this common dendritic cell progenitor that expresses CLEC9A, and so you can use CLEC9A Cre to label all the descendants of this progenitor. And so you can see here that we label dendritic cells, but we don't label TCs using this CLEC9A Cre fate mapper. And so that suggests that there's a unique progenitor that gives rise to these 4 different subsets, and that's separate work that's ongoing in the lab. But importantly, when thinking about the translational relevance of these cells, we found them not only in mice, but also in humans. And we originally reanalyzed the human gut atlas from Sarah Teichmann's lab, but actually I now show this slide because there was a much more exhaustive study that was done by Marco Colonna's lab after our study was published, in which he identified these raw gamma T dendritic cell-like cells in human lymphoid tissue, in particular within the intestine as well as the nasal mucosa. And interestingly, when he performed a transcriptional analysis comparing the phenotype of the human cells to the mouse cells that we had described, you can see that they have overlapping similarity with the 4 TC subsets that we had identified. Now, I'm only briefly going to touch on this just because I often get asked. When we published our study, we published in parallel with 2 other groups, one paper from Dan Lippman's group and one paper from Greg Sonnenberg's lab. And we all effectively showed the same thing, which is that you needed antigen presentation by these raw gamma T expressing cells to drive peripheral Treg induction. But unfortunately, we all called the cells different things, and so that has led to some confusion in the field. And really, one of the issues is that we didn't have the same number of cells in our different data sets. So we had identified 4 different subsets. In Dan's study, they had identified these Janus cells, and they identified 3 subsets. And then in this study, they identified 2 subsets that they called extrathymic air-expressing cells. So since all of this data has been published, that gives us the opportunity to now integrate all of the different data sets to see how these cell types align. And so what we did is we took each of the individual data sets, and then we extracted the cells that expressed both raw gamma T and MHC class II. And then we integrated the data, and this is the combined UMAP. You can see here that I've colored the cells according to the cells that we had identified. So we have these innate lymphoid cells, the ILC3s, which were the classical raw gamma T MHC2 expressing cells, and then these 4 subsets we had identified. Now, in the other data sets, what you can see is that there's obviously commonality. Every data set had this cluster of cells that expressed air that was called either JC1 or extrathymic air-like cells, ETAC1s, and they overlap with this subset TC1. But the other subsets of cells that had been identified actually turned out to be a mixture of TCs, either TC2 to 4 or TC3 and 4. And we think that the number of cells was probably the reason that the cells didn't separate into distinct clusters. And one reason that may have affected the abundance of the cells was the age at which the mice were profiled. So we had decided to analyze gut lymph nodes just at this time window when mice were weaning from milk to solid food, when the peripheral Treg cells were emerging, but the other studies had analyzed the mice a bit later, and obviously I showed you the developmental window for the cells, and they're actually starting to decline at these time points. And we think that this is important, not just from a nomenclature point of view, but really for being able to establish the function of these cells. So the 4 different subsets are transcriptionally very distinct, and we can see that it's this subset, TC4, which expresses key molecules that drive peripheral Treg differentiation, whereas the other subsets, which express programs related to the air-expressing MTEX, we think might have roles in tolerance to self-antigen. So the question we wanted to know more recently is, how do these cells regulate tolerance to other types of antigens beyond microbial antigens? And we know that different types of tolerance are mediated through these peripheral Treg cells, including tolerance to food, as well as fetal antigens. And so the one that we decided to focus on next was tolerance to food antigens. And the reason for that was really because of this interesting developmental wave where the cells are peaking just as mice are starting to encounter oral antigens within the intestine. So actually, the best evidence for an early life window for immune tolerance actually comes from the study of humans who are at risk of allergy to peanuts. So when I was doing my PhD at King's College, there was a study that was being conducted by the neighboring lab that was led by Gideon Lack. And what they had noticed was that the incidence of peanut allergy was actually on the rise in the UK. And if you looked in particular at Jewish children, they seemed to have a very high rate of incidence of peanut allergy compared to children in Israel. And that was thought to be because in the UK, the guidance had been to actually avoid peanuts and introduce them later in life. And that was thought to be a strategy which would actually prevent peanut allergy. In contrast, in Israel, children are fed this snack called bombus, which actually has peanut in it. And they receive that at a very early age. It's almost one of the foods that the children get weaned onto. And so it was thought that maybe this early life exposure to peanut allergy was actually promoting tolerance. And so they did this clinical study where patients were randomized to either early introduction of allergen or allergen avoidance. And you can see that there's this striking reduction in peanut allergy, both in non-sensitized and sensitized individuals, in those that received this early peanut exposure. And so we wondered whether this was because of these cells that we had identified that were providing this early life window for instructing peripheral Treg generation, and that if you time the antigen encounter with the development of the cells, you're more likely to achieve immune tolerance. And so 2 members of the lab set out to answer this question, Yolanda and Vanya. And first of all, they took an unbiased approach, and they decided to gavage mice with fluorescently labeled ovalbumin, so egg protein, and then see which of the antigen-presenting cells in the lymph nodes actually take this up. And so you can see that what they did is they gavaged mice that were 14 days of age. So this is the age when mice are just starting to eat food. And you can see that when they looked in the lymph node 16 hours after the gavage, there was only really prominent uptake by 3 cell types, and that was the TC4 subset, this migratory dendritic cell, which we heard about, CCR7 DC1s, and then a very small fraction of CCR7 expressing DC2s, which you can also see have less of the fluorescent label on a per cell basis. And that might be relevant to what we heard from Pierre about how they may actually have less antigen and therefore drive less TCR signaling, and I'll come to that later in the talk. So the next question we obviously had was, what's the functional role of this antigen presentation by the different antigen-presenting cells, starting with TCs? So to address this, we went back to the mouse model where you delete MHC class II on the raw gamma T expressing cells, and then we use the classical model of tolerance induction to ovalbumin, where you transfer these naive T cells that recognize the ovalbumin protein, and then you expose the mice to ovalbumin in the diet. And again, we adapted this for young mice, so we transfer the cells at day 14, and then we gavage the mice with ovalbumin, and we also put ovalbumin in the drinking water, and then we analyze the mice 1 week later. So what you can see is that we can find these congenitally marked OT2 cells within the lymph nodes in both the wild-type mice and the mice that lack class II expression on raw gamma T. But when you look at the phenotype of these cells, you can see that in wild-type mice, you get this very prominent Treg conversion. Now, what's interesting is that in contrast to the setting in the microbiota, where all of the microbiota-reactive T cells were said to be raw gamma T positive, you can see that in the dietary setting, it's a little bit different. About half of the Treg cells express RORgamma T, and the other half are RORgamma T negative. And at the moment, we don't know the significance of these two different populations, but they're both peripherally induced Treg cells. Now, what's very striking is that when you delete class 2 expression on the RORgamma T expressing cells, you completely lose both populations of Treg cells, both within the lymph node and within the small intestine. Now, what happens to these cells is instead of becoming Treg cells, they become effector T cells. Some of them become Th17 cells, but really the main response you get in mice on B6 background is that they default to this Th1 program. Now, this obviously isn't your typical allergic response because that's normally driven by type 2 immunity. And that's been noted before that B6 mice are not particularly prone to Th2 responses. And so we turned to a model of type 2 hypersensitivity that had been described by Daniel Musida's lab. And what you do is you immunize mice with ovalbumin conjugated to alum, which drives this Th2 response. And then you can challenge mice in the airway intranasally with ovalbumin and then look in the bronchoalveolar lavage fluid to look at the immune response, and you can also measure the antibody response. Now, what happens is if you pre-feed mice with ovalbumin, you actually protect mice from this inflammatory response. So you can see here in wild-type mice that receive ovalbumin in the diet, you have this reduction in eosinophils in the bronchoalveolar lavage, and you also see that you have this absence of antibodies against ovalbumin. However, if you delete class 2 off the RORgamma T expressing cells, you have this eosinophilia, and you still have antibodies against ovalbumin, even though they've been pre-fed with the ova. So this shows that there's a very important role for these cells in regulating oral tolerance. So we had previously profiled these cells by single-cell sequencing within the gut lymph nodes, and we wanted to understand whether it was the same subset, the TC4 subset, that was driving tolerance in the dietary antigen setting, and also wanted to understand whether the phenotypes of TCs were conserved across different lymph nodes. And so what we did is we sorted TCs from individual lymph nodes, both gut-associated lymph nodes as well as non-gut lymph nodes. You can see here that we have the same 4 clusters of cells that we identified previously, so TC1 to 4, as well as these early proliferating cells, and they express the signature genes that we had shown previously. Now, just highlighting here the expression of 2 important molecules that have been shown to drive Treg differentiation. One I mentioned is integrin beta-8, but another is this enzyme RALDH2, which generates the active retinoic acid, which has also been shown to be important for Treg differentiation within the intestine. Now, what was very interesting is when we looked at the TC composition according to the lymph node that we had isolated the cells from, what you can see is that in dark purple, this is the TC4 subset, we see that the TC4s are specifically enriched within lymph nodes that drain the gut or gut-associated lymphoid tissue, such as Peyer's patches. But outside of the gut, we have very low proportions of TC4. And we think that becomes relevant as to why the gut is such an important site for peripheral Treg generation. But if you encounter antigens at other sites, for example, through the skin, it's been known that you're more likely to generate an inflammatory or inflammatory response or become sensitized to those antigens. And this isn't just a difference in proportion that may happen in single-cell sequencing. So we were also able to show with this integrin beta-8 reporter mouse, where we could now identify TC4s by flow, shown here. These are the CD11b integrin beta-8 expressing TCs, that when we quantify these cells across the different lymph nodes, you can see again that we really don't find any of these cells within the skin-draining lymph nodes, but they are present within the gut lymph nodes. Okay, so then the hypothesis was that it was integrin beta-8 expressing TCs that were driving these food-specific Treg cells. And so to test that, we could ablate integrin beta-8 on RORgamma T expressing cells and then look at the tolerance induction when we transfer these ovalbumin-specific T cells. And again, what we find is we completely lose these peripherally induced Treg cells in these mice. So in the setting of food antigen, we can now ask something very interesting. So one of the things I always get asked is, well, what happens if you encounter your antigen outside this early life window? Now, importantly, you still have TCs. It's not that they disappear after this early life window. You just have a lot less of them. And so then we thought, okay, what we can now do in this dietary antigen setting is that we can actually now introduce the antigen in adult mice and then see what happens if you have no antigen presentation on TCs. And so to do this, we take advantage of this raw gamma tamoxifen-inducible Cre allele that allows us to temporally ablate MHC2 expression on the cells. So we take adult mice, and then we put them on this tamoxifen diet for 2 weeks. And then we transfer the ovalbumin-specific T cells, and then gavage the mice with ovalbumin and leave them on the drinking water. Now, in this setting, you can see here, these are your peripherally induced Treg cells. We still lose the Treg cells, or we don't generate them if you don't have antigen presentation on these raw gamma-expressing cells. But I think that what you can appreciate is that the proportion of Treg cells that you generate within the lymph nodes is far less than when we transfer OT2 T cells into the 2-week-old mice. And we think that that balance of Treg cells to T effector cells is very important in determining the degree of tolerance and may be important for determining how easily you can break tolerance. And so we're looking at that at the moment in terms of how easily you can sensitize the mice to ovalbumin according to whether you encountered it in early life versus adulthood. So Yolanda had a baby when she was doing this paper, and she was so impressed with the results that she enrolled her child in her own version of this oral tolerance study. This is her eating her peanut butter when she was starting to be weaned. So that brings us back to the question then. I showed you at the beginning that there's antigen uptake by dendritic cells as well as Thetis cells. And so the question then is, well, what are the dendritic cells doing in this context? And so to answer this question, we can now develop more specific models for targeting DCs now that we know specific genes are expressed by either Thetis cells or dendritic cells. And I showed you at the beginning of the talk that one of the genes that is uniquely expressed by classical DCs during their development is this gene, CLEC9A, which we also heard about in today's talk in the morning. So what we know about DC1s is that they're dependent on this transcription factor, IRF8. And previously, it was suggested that DC1s were actually the tolerance-inducing subset. And that was because if you delete IRF8 on using this other DC driver, ZBTB46, you lost peripheral Treg induction in the setting of oral tolerance. However, what we now know is that TCs also express ZBTB46, and they also express IRF8. So we first decided to generate a more specific model for depleting IRF8 in DCs using a CLEC9A Cre. And with this mouse, you can see that we effectively delete all of the DC1s, and that's both within lymph nodes and within the tissue. And now when we look to see what happens when you try and establish oral tolerance in these mice, you can see that they generate Treg cells without any issue, which is obviously what we would expect. But interestingly, what happens instead is instead of generating these Th1 effectors in response to the food antigen, you can see that these Th1 cells are down both within the lymph node and within the lamina propria. So that highlights this division of labor whereby the classical DCs are actually driving Th1 differentiation and the Thetis cells are driving Treg differentiation. But that also gave us an interesting clue because I mentioned before that if you delete IRF8 in the ZBTB46 expressing cells, you lost peripheral tolerance. So now we started wondering, okay, well, is there a role for IRF8 in the differentiation of TCs? And indeed, when we went back to our RNA and attack sequencing data, what we could find is that as well as expression of IRF8 within TCs, there was also enrichment for IRF8 activity as predicted by our chromvar analysis, which suggested that there might be a role for IRF8 in the differentiation of these cells. And so what we've done is we've deleted IRF8 of raw gamma T expressing cells, and then we look to see what happens in the context of oral tolerance, and you can see that we very nicely lose these peripheral Treg cells, both within the lymph nodes and the lamina propria, and so we're now currently looking at the impact on TC differentiation. It certainly affects their numbers, but we're also looking to see whether it qualitatively affects the cells that are left over as well. So in the last few minutes, then I'm just going to turn to then what's the role of antigen presentation on the DC2s. And so to do this, when we started these studies, we didn't have a specific model for targeting the DC2 lineage. So we instead decided to use a pan-DC targeting approach, knowing that we already know what the phenotypes are when we target DC1s. And so to do this, we took advantage of the fact that CLEC9A is expressed by the progenitor. And so if you delete class 2 expression using a homozygous CLEC9A Cre allele, you can actually target both DC1s and DC2s. And I'm just showing here that, again, if you use a homozygous Cre, you can label up to 70% of your DC2s, and obviously the DC1s express CLEC9A, so they're labeled. But we don't see significant labeling within the TCs. And so we can use this to delete class 2 expression on both the DC1s and the DC2s, and now it becomes very interesting. So if you look to see what happens to the OT2 T cells in this setting, by proportions, there's no impact on peripheral Treg cells. They obviously have this reduction in Th1s, which we attribute to the DC1s. Again, the Th17 cells aren't affected, suggesting that Th17 differentiation is not controlled by a classical DC in this context. But when you start to look at the actual numbers of the cells within the lymph nodes, you can see that in every single lineage that we look in, we have this a much lower number of both Tregs and T effectors. And so then we sort of thought about what was happening in this model, because we had shown that you didn't need antigen presentation by DCs to induce, for example, Treg cells. And we thought about the fact that we've got this chronic antigen exposure because we were actually leaving the mice on the drinking water for the full 7 days. And in contrast, previous studies often just do one gavage of ovalbumin and then look later. So we went back and we started to do a time course analysis. And what we found is that if you gavage the mice with ovalbumin and then look 48 hours in the lymph node, we don't see any phenotypes. The OT2 cell numbers are equivalent. All of the different lineages are starting to arise. But if you go 1 day later and look at 3 days, you can now start to see that in every lymph node that we look in, there's this reduction in OT2 cells. And interestingly, when we look at this time point at 72 hours, the cells that are there have upregulated CD44, so they're antigen experienced. But when you look at, according to cell division, you can see that as you go through rounds of cell division, this is when you have this reduction in OT2 T cells becomes apparent. And so our current working model is that you actually need sequential interactions with antigen-presenting cells. So you first have to prime your CD4 T cell response, and we think that that's either driven by TC4s for regulatory T cells or, for example, DC1s for Th1 cells. But then you need this second interaction from a dendritic cell, which we think is a DC2 because we didn't see this phenotype when we just ablated the CCR7 expressing DC1s. But obviously it could be that you need both DC1s and DC2s for this. And one possibility is that the DCs are interacting with the mature effector T cells to sustain them, but another possibility is that they act through a stem-like progenitor to maintain this reservoir that then continues to output effector T cells. And this is what we're currently exploring at the moment. And although this is obviously in the setting of dietary antigen, we think that these rules that we're starting to uncover for dendritic cells will apply in the settings of other chronic antigen stimulation, for example, during tumor formation. So this is our current understanding of the antigen-presenting cell landscape in addition to these classical DCs that we had identified previously. We now have this additional lineage of antigen-presenting cells, and we think that these different antigen-presenting cell subsets instruct different aspects of the immune response. So I've shown you that the DC1s were regulating Th1 responses. I didn't show this to you today, but we found that the DC2s are regulating the TFH response, and of course previous work has already described a role for these CDC2B cells in regulating Th2 cells, and now we have this lineage of antigen-presenting cells that includes this Treg-inducing subset. So I highlighted that we think that there's something quite interesting that goes on in early life where you have this wave of cells. And so that suggests that there may be particular cues according to the lymph node that drives the differentiation of these cells, and in particular, this TC4 subset. And so this is work that's ongoing. We're looking at cues that are provided through the breast milk as well as the microbiota, and also unique cell populations that are enriched within these early life lymph nodes. But I think that what this highlights very nicely is that you really need to think about the composition of your lymph nodes when thinking about the type of immune response that you're going to elicit. And I'm sure Wolfgang is going to go into this in far greater detail. But I think what you can now really appreciate is that if you have tumors that, for example, drain to gut-draining lymph nodes where you have this enrichment of tolerogenic cells, there is a possibility that actually the outcome that you're going to generate is more of a tolerogenic immune response versus an inflammatory response that would be driven by these classical dendritic cells. I hope that one thing I've also highlighted is that these cells have a developmental wave, but I don't want the take-home message to be that therefore this is only something that is important during early life, because one of the things that we found is that certain cues can actually lead to the expansion of TCs, or perhaps a new wave of TCs within lymph nodes. And one of those cues, interestingly, turns out to be tumor formation. So this is looking in an orthotopic model of melanoma where you implant B16 cells. And we've done this both in neonatal mice and in adult mice. And this is just some initial data. But what you can see is that we look to see which cell types are enriched in the tumor-draining lymph node relative to the non-tumor-draining lymph node. So anything above 1 is enriched. So of course, you can see a lot of immune cells are enriched within the tumor-draining lymph node. But what's very interesting is that we have this really striking enrichment of TCs within the tumor-draining lymph node, in particular within the adult mice. And so that suggests that the tumor may actually co-opt these tolerogenic immune cells to suppress immune responses, and that's work that's ongoing in the lab at the moment. So I'm going to end by acknowledging everyone in the lab. I sort of mentioned people who have been involved with the different projects along the way, but it's very much a team effort. And we've also had really amazing collaborators, both on the computational side at Sloan Kettering, as well as collaborators at the Ricken, where we were studying the development of the cells, and of course our funders.